Hello Year 12, welcome to our next video. We are exploring a concept that we kind of touched on in our last video, which was this idea of what an enzyme was, and I said that they were a molecule that was involved in metabolism. So today we're looking at 9.2.1.1, identify the role of enzymes in metabolism, describe their chemical composition, and use a simple model to describe their specificity on substrates. Let's get stuck into it. So, first job is to unpack this dot point. What do we need to do? There are three things we need to do here today. Firstly, we need to identify the role of enzymes. What's their job? Alright, and what's their job in relation to metabolism? We need to describe their chemical composition, so we need to describe some features of their chemical composition. And then, this is an interesting one, it crops up a couple of times in the syllabus. We need to use a simple model. So we've got to figure out a model of something, and then we're going to use that model to describe, which is get some features of their specificity on substrates. Now, we haven't come across substrates yet, but we will. Let's get into it. First off, we need to have a couple of definitions down pat. Metabolism, if we remember from our last video, is basically any chemical reaction that happens inside of the cell. Alright, if it's required for life, if it's a reaction for life, we can whack it under the heading called metabolism. Okay, so the next one is that we need to have a look at enzymes. So an enzyme is a protein. All enzymes are proteins, but not all proteins are enzymes. So they're a part of the category of proteins. Alright, and they act as this thing called a biological catalyst. That's what we're going to be exploring in a moment. Alright, so basically, we are looking at this idea that without enzymes, the reactions that we need for life would be occurring far too slow in order to support it. So basically, enzymes speed up the reactions inside of cells. So this is normally like just stupidly fast. Like we really can't. We can, but we really can't get a grips on appreciate how quick this speeding up is. It's pretty impressive. And the cool thing is the enzymes are not used up. Once we make one enzyme, we can reuse it over and over and over, which is pretty cool. Alright, so looking at this role of catalysts, I had two diagrams here. This is the one that we're going to see most commonly in a textbook or in an exam paper. And basically, this is just that if we didn't have an enzyme, our blue line here represents the amount of energy it would take, alright, in order to release the energy. Alright, so we started with some products, we started over here. In order to release the energy, which is this stuff here. Release this stuff here. If we didn't have an enzyme, the blue line is basically if we had an enzyme facilitating this chemical reaction, the activation energy is far less. So it's kind of like, look, you pay me $1. And I promise you the return on that dollar is going to be $5. And this one here is, hey, you pay me $4 and I promise you the return is $5. So basically you're getting more bang for your buck here. Okay, we are using less energy in order to facilitate a chemical reaction. So that's what's shown here. Over here on this diagram, I like this one because it's got a very nice 3D image, and it kind of shows you the blocking issue. Alright, so it's kind of same concept. Blue line is this big one. Alright, and there's a wall. If you don't have the energy, you can't create the reaction. Okay, you have to actually have enough energy to get over the hill. But if you lower the hill, you obviously only need a small amount to get to your product over here. Alright, so hopefully you guys understand what a catalyst is now. They speed a reaction up and they reduce the energy cost of that reaction. That's a pretty important thing for life. Alright, so in general, proteins are large and that means they kind of are three-dimensional. So Three dimensions is a pretty important concept to have because three-dimensional objects have a shape. A protein here is a polypeptide chain. All right, so a polypeptide chain is an amino acid. We often hear about amino acids being important in bodybuilding, and it is because these amino acids, we eat a steak, we digest the protein, we chop the proteins up into their basic building blocks, which are amino acids, and then we link them all up together again. And for bodybuilders, they do that so that they can repair their muscle proteins a lot faster. But they're connected with this thing called a peptide bond. So anything that is a chain of molecules together, tied together with a peptide, they turn into poly, meaning many, and peptide, meaning the type of bond that holds them together. Alright, so generally these enzymes usually have a name ending in ase, such as amylase. Amylase is a pretty common one that we come across, and it's an enzyme found in the mouth. Amylase is used to digest starch. It chops the long chains of starch molecules up into smaller, simpler sugars. It's kind of like why if you have bread in your mouth, which is very starchy, if you leave it in there and coat it in saliva, it's likely going to start tasting sweet after a little while. Alright, so going back to this shape, this three-dimensional globular shape contains a functional area. So not the whole thing, but there is an area on that protein, on that enzyme, that's called the active site. And this is the active site is this kind of little zone, little surface, where if a substrate bonds with the enzyme can perform its magic on it. And by performing its magic, we mean like kind of force two molecules together or rip two molecules apart, depending on what the job of the enzyme is. Alright? And that last dot point just says that too. Alright, so because of that shape, you've got to think of it, every surface has its own shape, so this kind of makes them specific, and depending on what shape they have, or what the shape of the active site is, this will determine what type of substrate can bond with it, and therefore what type of substrate the enzyme can act upon. In order to explain this, scientists like to use models, and we're going to explore two models, and hopefully they'll help you understand this concept of what an enzyme does. The first one is a lock and key model. When we immediately think of lock and key, you go, right, okay, the key fits into the lock, and if it's the right key, it opens up. And that's a kind of good way to start this conversation. So we've got this enzyme, and over here we have two substrates coming in, and you can see that these two substrates, when they line up, they'll match the active site here, which is a pretty important job. Alright, so these enzymes can act upon two different substrates, and they form this thing called a enzyme substrate complex. This is what happens here. You can't just go from here to here and go, yeah, my model's awesome, because you haven't actually explained that the enzyme's job is to fuse this little line here together. Alright, and it does that by placing physical and chemical stress on the molecules. What it makes is a new product, so we label this with P. So we started with S, and if you've got, you could have S1, S2, all the way up to S15 if you wanted to. It doesn't matter what that number is. You can fuse them together and you make a product. Now, what's cool with this model is that if we were to cut these out, you would notice that you could reuse the enzyme. Yay for not being used up in the process. The next model that we look at is a very similar version of the lock and key model, but it's a bit different. This one it doesn't quite have the same shape. And kind of, what does this mean? Well, this is the idea with this model, is that an enzyme can kind of change a little bit in order to fit the appropriate substrates. This does make them a little bit less specific, but in terms of reality, we've actually found out that enzymes can kind of change it up a little bit and kind of be not just specific to one enzyme, but maybe 2 or 3. And enzymes can be specific to one substrate, but can be specific for more than one, 2 or 3. Anyway, so here we go. The induced fit. The substrates bind to an active site, and the binding induces a temporary change in the shape of the enzyme. This is this concept of inducement. A chemical reaction occurs, the substrate is changed, and the products are released. And again, the enzyme is not used up in the reaction. And this can go both ways depending on the enzyme. So you might need a different enzyme to break P down into two new products. But what this is saying is basically that there are enzymes that will fuse 2 molecules together or break a large molecule apart. Right, so there's our 2 models. Let's just have a look at them. And whenever we talk about models in the course, this is going to come up frequently in this course, so we just need to be aware of it. Whenever we use a model, we've actually got to go, was that model helpful at all? Because if it wasn't, then it's a pretty crappy model. So we want to try and use models to explain concepts. So the first one we used was lock and key, and alright, so its advantages were, look, it gets us understanding enzyme specificity. As soon as you say lock and key, you kind of go specific lock, specific key, hey presto, specificity. Alright, that's pretty cool. So it simplifies a biological process, and we can't see it. Alright, even though they're 3D, they're tiny little molecules interacting at ridiculous speeds in our body. So lock and key model is good for understanding specificity and showing a process that we can't actually see with a light microscope. But, models always have some flaws, so let's look at the flaws. Alright, so this one is, it's considered less accurate than the induced fit model. And when we were looking at this model, we actually had no idea of how much time it took for the substrates to create and the enzyme to create the enzyme substrate complex, and then to produce the product. So, what the hell? We're now kind of lacking in this piece of information. But that's okay. We just now need to go. All right. Well, we can steer. Could we speed this up, or could we put a timer to it? It's okay with that, so long as you understand what the limitation was. The next one is the induced fit model, and we like this one. The advantages, it's similar. We kind of get an idea of specificity, but what we get most is a better picture of reality, and that's kind of cool. When a model reflects reality, we like it. All right, so it's the most accurate model of enzyme activity. It reflects reality the most. All right, so the limitations on this one is that it is supported by evidence, but it is there is evidence to say that maybe the lock and key model is better, but we think at the moment the induced fit model is a good one. So we haven't kind of nailed it to one model. That's why we can talk about 2 of them. And again, similar to the first one, it lacks a time frame. All right, so that's the dot point wrapped up. Let's have a look at our summary. First off, enzymes are proteins that catalyze metabolic reactions. They're large with unique shapes. Their active site defines the substrate that they can act on, making them highly specific. Models can be used to represent this specificity. We looked at 2, the lock and key model and the induced fit model. Alright guys, thanks for listening. Cheers.